Hi everyone, welcome back to my channel. It's me again, Jessica. So our topic for today is all about microbial genetics. Microbial genetics, the study of how genes are organized and regulated in microbes in relation to their cellular functions, closely related to the field of molecular biology. Structure and functions of genetic material. DNA structure. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA's instructions, or genetic code. Nucleotides are attached together to form two long strands that spiral to create a structure called a double helix. The bases on the strand pair with the bases on another strand. Adenine pairs with thymine, and guanine pairs with cytosine. DNA molecules are long that they can't fit into cells without the right packaging. To fit inside cells, DNA coiled tightly to form structures we call chromosomes. Genetic material changes by the process of mutation. It carries all the important information which is needed for the proper functioning of the cells. The genetic material is capable to replicate and inherent to the offspring. Genotypes are the genetic makeup of an individual. Phenotypes are the physical traits and characteristics of an individual and are influenced by their genotype and the environment. DNA and chromosomes. DNA, deoxyribonucleic acid, is the cell's genetic material contained in chromosomes within the cell nucleus and mitochondria. A chromosome contains many genes. A gene is a segment of DNA that provides the code to construct a protein. The DNA molecule is a long, coiled, double helix that resembles a spiral staircase. The flow of genetic information. Genetic information is stored in the sequence of bases along a nucleic acid chain. The bases have an additional special property. They form specific pairs with one another that are stabilized by hydrogen bonds. The base pairing results in the formation of a double helix. A helical structure consisting of two strands. These base pairs provide a mechanism for copying the genetic information in an existing nucleic acid chain to form a new chain. Genes specify the kinds of proteins that are made by cells, but DNA is not the direct template for protein synthesis. Rather, the templates for protein synthesis are RNA. Messenger RNA ribonucleic acid molecules are the information carrying intermediates in protein synthesis. Other RNA molecules, such as transfer RNA and ribosomal RNA, are part of protein synthesizing machinery. All forms of cellular RNA are synthesized by RNA polymerase that take instruction from DNA templates. This process of transcription is followed by translation, the synthesis of proteins according to instructions given by mRNA templates. Thus, the flow of genetic information or gene expression in normal cell is. This flow of information is dependent on the genetic code, which defines the relation between the sequence of bases in DNA or its mRNA transcript and the sequence of amino acids in a protein. DNA replication are the same in prokaryotic and eukaryotic organisms. Also, the biological process in which two identical copies of DNA are produced from one original DNA molecule. The first step in DNA is to unzip the double helix structure of the DNA molecule. RNA and protein synthesis. Messenger RNA provides the ribosome with the blueprints for building proteins. Amino acids are the building blocks of proteins. Each amino acid in a protein is delivered to the ribosome by yet another type of RNA, transfer RNA. Transcription in prokaryotes. The process of synthesis of RNA by copying the template strand of DNA. During replication, entire genome is copied, by in transcription only the selected portion of genome. The enzyme involved in transcription is RNA polymerase. Unlike DNA polymerase, it can initiate transcription by itself. It does not require primase. More exactly, it is a DNA-dependent RNA polymerase. Translation is the process that takes the information passed from DNA or messenger RNA and turns it into a series of amino acids bound together with peptide bonds. It is essentially a translation from one code nucleotide sequence to another code amino acid sequence. The ribosome is the site of this action, just as RNA polymerase was the site of mRNA synthesis. The ribosome matches the base sequence on the mRNA in sets of 3 bases called codons to the RNA molecules that have 3 complementary bases in their anticodon regions. In eukaryotes transcription, eukaryotic transcription is the process by which the information in a strand of DNA is copied into a new molecule of RNA. It is the first step of gene expression, in which a particular segment of DNA is copied into RNA, especially mRNA, by the enzyme RNA polymerase. RNA processing eukaryotic cell. RNAs are made in the nucleus of a eukaryotic cell, but function in protein synthesis in the cytoplasm. RNAs undergo extensive modifications after synthesis by RNA polymerase II. Transcription and translation. Transcription in the nucleus, the cell's machinery copies the gene sequence into messenger RNA, mRNA, a molecule that is similar to DNA. Like DNA, mRNA has 4 nucleotide bases, but in mRNA, the base uracil replaces thymine. translation. The protein-making machinery, called the ribosome, reads the mRNA sequence and translates it into the amino acid sequence of the protein. The ribosome starts at the sequence AUG, then reads 3 nucleotide codon specifies a particular amino acid. The stop codons UAA, UEG, and UGA tell the ribosome that the protein is complete.